In this part of the video, we will be understanding the development of a dicot embryo, and this is a part of post-fertilization event. That means after the fertilization process, that is the fusion of the male gamete with the female gamete, now we are going to study the development of dicot embryo. The structure or the formation of the endosperm has already been dealt with. Now let us come to the process of development of an embryo. The process of development of the embryo is known as embryogenesis, and we are going to study under two heads. That means the process of development of embryo in a dicot plant and in the case of monocot plant. So first we'll be dealing with dicot. Now just remember the process or the sequence of events that has taken place during the fertilization. During the fertilization process, this is the embryo sac, and the zygote formation has taken place after the fusion. And remember, the zygote is a diploid structure. So we have already labeled over here the micropyle end and chaliceal end. It is important for our reference, and the structure then becomes very clear. So always label with the micropylar end and chaliceal end. Now we all know that due to the fusion of the male and the female gamete, the zygote has been formed, and this zygote is a diploid structure. As soon as the zygote formation has taken place, the synergid starts degenerating and also the antipodal cells. Here we have the development of the nucleus, that is the primary endosperm nucleus will finally forms the primary endosperm cell, and they are a triploid structure over here. Now for this development, we'll be focusing mainly on the zygote. Now the zygote, which is present towards the micropylar end, undergoes mitotic division. But this development, always remember, that is the development of the zygote, will take place only after certain amount of endosperm formation has taken place, because this is important. As the endosperm formation has taken place, the nutritional material will be provided by the endosperm. So the zygote will have sufficient nutrition as it will develop from one stage to another stage or during the formation of embryo. So the development takes place towards the micropylar end, and only after the certain amount of endosperm formation has been done. Till then, the zygote will remain at rest position. That means it will not divide. So after the zygote has divided, it will form two cells. Now the two cell which is towards the basal end, the cell which is towards the micropylar end is known as the basal cell, whereas the cell which is towards the chaliceal side is known as the terminal cell. So we have unequal division, and which results into a terminal cell and the basal cell. Now we'll study one by one how these two cells divide. The basal cell, it undergoes transverse division, and due to the transverse division, that means the cell will divide like this continuously, and it will form a structure like this. These are the divisions which we are focusing. I have labeled it as black over here, and this black cells, that is the basal cell, will divide to form a structure and a large rounded structure which is known as posterior cell. These cells, which are due to the division, transverse division, and also remember all the divisions which are taking place in the zygote are all now mitotic divisions. So it will undergo transverse mitotic division to form a sequence of cell which is known as suspensor, and a larger cell that is known as posterior cell. This is towards the micropyle end. The suspensor helps in pushing the developing embryo towards the endosperm, that is pushes deep inside the endosperm. So if the zygote is there, then the formation of the suspensor will push the zygote or the developing embryo towards the primary endosperm cell, that is the endosperm which develops finally. Now, coming to the terminal cell. The terminal cell, the division will be taking place in all the planes. So, their division will be taking place in all the planes, and due to which it will appear a rounded mass. And this stage is known as pro-embryo stage, because from here now the cells will multiply further to develop the embryo. So the pro-embryo, and initially it will divide in all the planes forming a mass, and the fore structure is known as pro-embryo or pro-embryonal quadrant. Now, so what is the stage we are having now? The terminal cell divides in all the planes to give rise to a rounded globular structure which is known as proembryo, and it is a mass of cells. We are able to see cell from one side only. Then we have the basal cell. The basal cell divides to form the suspensor and the haustorial cell. The function of the suspensor is to push the developing embryo inside the endosperm, and haustorial cell holds on to that, and this is the structure which has undergone or which develops from the terminal cell and the basal cell. Coming to the next step, the proembryo divides further, but remember there is no change in the suspensor. That means the cells in the suspensor will not divide further, but the proembryo will continue to divide. Now see, the development has to be from the proembryo to the embryo level. So the division will be taking place over here. Now the proembryo will divide further and result into a proembryonal octant. Again, this is a mass of cells. Since the division is taking place in all the planes, it will appear as a mass of cell, and this is now known as octant, because when we look from one side, it appears to have 8 cells, and it is a rounded structure. There is no change in the haustorium and no change in the suspensor, but due to the increase in the size of the proembryo, we call it, or proembryonal octant, the suspensor seems to be squeezed, but there are no division. Also, over here, you find there is a cell which is towards the proembryo. This is the proembryo, and there is a cell which is marked in red over here. This is the cell which is towards the proembryo. First cell, this is known as hypophysis. Now each of the cell will divide to give a particular structure. Coming to the next part, when we study the division, in the proembryo, till here the divisions are taking place in all the planes. Now the division in the case of proembryo, they will be periclinal division. We are focusing on the development, how one-sided, that is proembryo, how it will divide. Then next on we'll discuss about the other structures, what role do they have. So the proembryo, it has now the periclinal division. This periclinal division, that means there is an inside and outside. So there will be formation of a structure which is these cells. That means these cells, if I draw like this, one, two, three, 4, 5, 6, 7 and 8, and these cells will have periclinal division, that is outer, inner cell, outer cell, inner cell, outer cell, inner cell. Like this it will have, and due to which we obtain a structure like this. Now this outer layer will form the epidermis. The structure, that is the epidermal layer, will be formed by the outermost layer which is formed due to the periclinal division. Now, the hypophysis undergoes division to form 8 cells. Now, since it forms 8 cells like this, over here, it is hypophysis, we have used a red color marker. So, here it divides to form the 8 cells. And there is no change again in the suspensor and the hostorium or the hostorial cells. So, there is no change. Now, let us study how the structure or which part of the plant develops from these cells. The suspensor and the hostorium will remain as such. There is no change over here. Then coming to the proembryo that results into the periclinal division, as told, the outer layer will develop epidermis. Now, when we look over here, the lower half, that is one which is towards the micropylar end, the lower half division is shown like this. It will develop hypocotyl. So the lower part which is towards the hypophysis will develop a hypocotyl, and the part which is above, this part will develop the plumule and the cotyledon. So from the proembryo, the part which is towards the chalazial side, that will develop the plumule and the cotyledons, whereas the side which is towards the hypophysis will form the hypocotyl. Coming to the hypophysis, hypophysis has undergone division to form 8 cell structure. Now, out of this 8 cells, the one which is towards the proembryo, the 4 cells, that is these two, they will form the root cortex. The cortex of the root will be formed by these cells which are present towards the proembryo side. The other 4 cells which are towards the suspensor, they will form the root cap and the root tip. So here, the radical part develops from the hypophysis. The root cortex will be developed from the cells which are towards the proembryo, whereas the root cap and the root tip will be developed by the cells which are towards the suspensor. Now, coming to this structure further, this part continues to divide. The proembryo continues to divide and it will form cells. It will be filled with cells. Now these cells, the cotyledons will swell up like this. The proembryo, the two parts will swell up like this and it will have the cells. Now, this part, since it is looking a heart-shaped structure, we call it as a heart-shaped embryo. Here you can see the two parts have swollen up and this will develop later as cotyledons. Now here, again, the hypophysis, that is the area, it undergoes the divisions and as a result, the root tip is formed and the other structure, root cap and root cortex is formed due to the further divisions in hypophysis. Here, due to this development, continuous development, this cotyledon will develop as this, and this cotyledon, the black, this one, will develop as this. And it cannot go erect because the embryo is pushed towards the endosperm due to suspensor and there is no space over there. So they appear bent like this. And this is known as horseshoe-shaped embryo. So when we study the development of the embryo, if the order is asked, the order is the globular embryo, it is changing into heart-shaped embryo followed by the horseshoe-shaped embryo. Now these two will be developing the structures over here. So they will be developing as cotyledons. This is the root part and the root cap and the root tip over here. Let us now come to the structure of the fully developed cotyledonous seed, dicot seed. Now when we see a dicot seed, there has to be two cotyledons. So we see over here there are two cotyledons which are developed from here. Then there is a structure which is seen as red over here. This is the structure which is seen as red over here. The two cotyledons meet at a point in that. And this, the two cotyledons where they meet is known as the cotyledonary node. Now, you can see over here the axis. This is known as the embryonal axis, the entire structure. Now, the tip which is towards the chaliceal end will form the plumule. As you can see the tip over here, this tip which is towards the chaliceal end will form the plumule, whereas the tip which is towards the micropyle will form the radicle. Now, here, between the space between the cotyledonary node and the plumule is known as epicotyl. You can see the cotyledonary node meets at somewhat in the center of the embryonal axis. That is right from plumule to the radicle. So the space between the plumule and the cotyledonary node is the epicotyl, whereas the space between the radicle, that is the tip of the embryonal axis towards the micropylar end, and the cotyledonary node is known as hypocotyl. So here is a diagram of the cotyledonary embryonal axis, which is dicot having two cotyledons. This is the complete development of the embryo which takes place in a dicot plant. Also, it is important for you to understand the structure of the cotyledons and the embryonal axis, because this is important when we'll be studying about the germination of the seed. Thank you.